Abdullah, can you see my screen?
Yes, I see. I will
Okay, let me
I
put
will
on full
introduce
screen.
you and we can start. Okay.
Okay.
Welcome, ladies and gentlemen. Thank you all for joining us today. My name is Abdullah Rafi, and I will be your moderator for this lecture on a hidden diversity and host specific gut microbiomes associated with the honey bee and stingless bee from sub-Sahara African region. We have an exciting session ahead, and I am delighted to see such a vibrant audience here. Please join me in welcoming our speaker, Dr. Yosef. He NRC postdoctoral research scholar hosted in therapy lab. He received a PhD in genetics, University of Pretoria, South Africa at 2021. He interested in bee gut microbiota, and his current project is artificial selection of honeybee gut microbiome, impact on host longevity and microbiome dynamics. Prior to starting his postdoctoral research, he served as a research assistant in animal health and PhD research fellow in environmental health at the International Center of Insect Physiology and Ecology in Kenya. Uh, he developed the first bacterial chemotoxis fishing method used to screen and enrich environmental chemotactic bacteria using a glass capillary. We will have a devoted Q&A session the end, so please save your questions for that time. Thank you, Dr. Yusuf, for joining us today to MGBS, and please start your presentation.
Thank you, Abdullah, for that wonderful presentation. Can you see my screen clearly? Let me know. Please confirm. Can you see the screen?
It- It's clear, Dr. Yusuf.
Thank you. Good. Hello, everyone. As Abdullah mentioned, my name is Yosef Tola. You can call me Yosef in brief way. Uh, Yeah, he has said a lot of things. I don't want to repeat the introduction. I'm really interested in the bees gut microbiome. From my background, from my childhood, I grew up in the rural area, and my dad is a beekeeper, and I used to see whatever my dad was doing during his beekeeping, and I used to see also all the challenges, all the problems. And that's why I'm really interested to investigate on this area and to find, I mean, understand all the biology. The first thing is to understand the biology and also finding maybe a solution for the challenges which our beekeepers are facing across the world. Uh, I'm working, I have a very strong collaborators, and the first three peoples are my current postdoctoral fellow collaborators, and the last two people are my PhD collaborators, and they have been contributing a lot on my journey. I really appreciate them. Yeah, okay, sorry. So, in my presentation, I have listed here my outline. First, I'm take you through kind of a brief introduction about this study, and then after introduction, I will share with you my finding on the honeybee gut microbiome in Africa, and then I'll take you through my finding on the stingless bee species gut microbiome, and then I'll go through a kind of a brief summary and some recommendation on that study. Then finally, I will highlight or share with you brief information about my current project for my postdoctoral fellow. Good. Now, this topic is about the bees. in general, we have two bees here, the honeybees and stingless bees. But why bees? These bees are very important and very special in our life. That's the first question we raise in our brain. What the importance of these bees? Yeah, bees they play a lot of roles in our life, in humankind life, through the pollination. They play a lot of roles in the pollination, pollinating our plants, and especially about 85% of the plants are based on the bees pollination. And not only that, and 1 out of 3 of all the food we eat, all the crops we depend on, they are pollinated by the bees. And for example, in United States, more than 100 types of the crops are pollinated by the bees. And not only this one, in the developing country or in small scale farm areas, uh, the bees they play a role for the farmers, for the beekeepers as a income generation, especially in our continent in Africa. Most of the beekeepers are they are not doing the beekeeping for hobby. The other developed countries they are doing beekeeping for the hobby, but for our continent, most people they do for the income generation to help their families and to send their kids to schools to get money to generate money. That's what my dad has been doing, is still doing. So, in general, the pollination service, world or the pollination service contributes about 235 USD to 577 billion annually to the global crop production. However, our bees are facing a lot of challenges, and so people they are asking a lot of questions now. Our bees are the population is declining, bees are dying. Why is this thing is happening? And in 2000, around about 2006, this the colony collapse, the decline of the bee population, the honeybee population reported in north, especially in Europe, in Canada, in and in United States. It's because of a lot of factors. As you see from the first picture, the bees, they have very strong social interaction. They work together. So in such social interaction, where there is outbreak of things, where there is a challenge of the disease outbreak, they can easily transmit and it can affect the colony easily. So uh, for this problem, there are a lot of factors which are contributing to these issues. Like, for example, the pesticide we are using in our agricultural areas, the insecticides and then the pesticide, the herbicide we are using. And we are using for our agricultural to upgrade our agricultural production. Indirectly, they are affecting our bees. How? They are in different ways. These are pesticide can directly affect the bees and also indirectly can affect the bees. The indirectly way is that pesticides can affect the gut microbiome, which is very important in the bees. That indirectly affects the hosts. And other factors, habitat destruction. When there is a habitat destruction, like for the kind of for maybe for the construction or maybe kind of the destruction for the agricultural things, it can affect the bees. How? It can affect maybe their flowering and their food, so they cannot access the food. And the other thing is that like kind of nutrition deficiency. That means what I mentioned, maybe if there is a kind of shortage of the flowers and foods, it may affect their population. The other thing is that kind of the air pollution. Air pollution can also affect the bees. Bees are flying about 5 kilometers to get their foods. Now, if their memory or if their brain is affected while they are flying, they may not come back to the hive. That means they die outside there. The other thing is the global warming, which is also contributing the weather changing and seasonal changing. If their flower season is changing, it directly affect the bees. So here what we can see is, and if these factors are affecting, number one, directly they are affecting the bees, indirectly they are affecting their biological system, like kind of their gut microbiome, their spatial and symbiotic microbes, which is indirectly going to affect the host. So that's why I'm interested to look on the side of the symbiote, the gut microbiomes, how the gut microbiomes are interacting with the different factors which are affecting the bee populations. Now, they've got microbiome not only in bees, and also they are playing a kind of different role in other insects, in different types of insects. And like for example, in Drosophila, the microbes are playing a role to influence the mating preferences. And also in locusts, they are playing a role producing the aggregation pheromones for the locusts. You know, the way the locusts, they live, the way they fly together. So the symbiont can help them to produce the aggregation pheromones, which can help them to aggregate. And what do we know in bees? And what do we know the bees about the microbes, the symbiont, which are associated with the bees? According to this report, so far we have uh, about 9 phylotypes of the gut microbiomes which are associated with the honeybees. The and in from these phylotypes, 5 of them, the Snogresella, Glamella, Bifidobacterium, the Lactobacillus species, are specific only to the bees. You can only find them in the honeybees. You can't find in other organisms. And the other 5 species you may find in 4 species you may find in other species. So these 9 phylotypes, you can manipulate and play with them to study and to use a honeybee as a model. That's what makes honeybees special from other insects. So these microbes they are very simple and they are very highly conserved in the honeybees. Not only in the honeybees, also in stingless bees, the most dominant microbes are lactobacillus and the bifidobacterium. They are most abundant genera in the stingless bees. Each of these microbes, they have their own role in bees. And for example, they play a role in digestion, metabolism, and also they can play a role in the nutrition. In addition, we know that the host has its own immune system or immune function, but the microbes contribute or initiate the host immune system to respond to the disease and the other factors which challenge the hostess. In addition, they have a kind of also like Freshella prora, which is very important to protect against the different pathogens. So each of them, they are contributing different roles in the honeybees. So for my study, I'm focusing on the African honeybees and African stingless bees. So why these African honeybees are interesting? Because most of this study, the one I've showed you in the first slide, they are studies from the Europe on the other in Northern Americas. But we don't have a lot of information about the continent, the African continent on the honeybees and why the honeybees are very important in our continent. We have the African population which seems to be more resistant to pathogens and parasites than the Northern Hemisphere honeybees. In addition, we have the important genetic diversity in African continent. And for example, only in Kenya, we have more than 3 subspecies uh, honeybees. However, we don't have a lot of information about their gut microbiomes and how their gut microbiomes are interacting and what their gut microbes are contributing. And also, we have a lot of questions why the African honeybees are resistant to pathogens or parasites. What is supporting them? Is it the environment is supporting them, or is it the microbes associated with them is supporting them? Yeah, we can raise a lot of questions here. The other thing is that why the stingless bees are interesting. I'm really interested also in stingless bees. These stingless bees are most diverse group among the calculate bees. They are distributed in the tropical region and subtropical regions. As you can see from this map, they are distributed in these regions. And especially in our tropical in sub-Saharan Africa, it's the home to the large diversity of the stingless bee species. Especially if I give you example, one in Kenya, we have 6 genera, which are composed of the 12 species of the stingless bees. And the other important thing is with the stingless bees, they do not sting like honeybees. That means since they are not stinging, we could use them kind of for the crop pollination, especially even in greenhouses. Sometimes it is difficult to put the honeybees in the greenhouse for a pollination, but you can easily put the stingless bees in the greenhouse for pollination service because they are not stinging. I mean, you can, while they are there, you can do your own work. And they can produce the same way the honey produce and propolis and the wax with high medicinal values or high medicinal properties when we compare with the honeybees honey. So that's why these are the most important. In addition to this one, these bees are the diversified species, highly diversified, highly populated in the tropical regions. They have different system, the way they build their nets and the way they clean, the way they live in social interaction. It's very unique and they're very important species. So that means, and I was thinking that maybe they have a kind of, since they have a different unique features, they may do have the different features even with their microbes. That's why I started studying with our stingless bees in Kenya. Now, that's my brief introduction, the background for my study, for my both studies. Now, I'm be taking you through the honeybee, the gut microbiome, what I found from my study. So, when I started studying on the honeybee gut microbiome, since we don't have a lot of information in Africa, apart from the information we have the latitude or other continents, asked myself what are the bacteria associated in honeybee gut in Kenya or in Africa, because maybe they are similar with others or different, which we don't know. That's why I started with this question. So, to answer this question, I focus on Kenya on for my sample size, and I focus on the western region, western Kenya, and which is very humid, which is humid and sub-humid zone, and also I sampled from the central Kenya, which is a temperate zone, and I sampled from the coastal Kenya regions, which is a coastal lowland zone, and that's why I focused on the different geographical location to get a robust data to answer this question. So once I get the honeybees from the different region of Kenya, the second part was kind of cleaning them because I'm interested in the internal or the gut microbiomes. So that means I have to clean the outside microbes from the body of the honeybees. That's why this is the way I dissect the gut and get the clean guts from their stomach. And then for this one, I did a DNA extraction. And from the DNA extraction, I did a sequencing, genome sequence, 16S sequencing, targeting the V4 region, and analyzed for the diversity in and abundance using time, pipeline, and also ARC package. So from this one, what did I find? And I found the bacterial genera which are associated with the honeybees in Kenya. Here is the result for the microbes which are associated. So the first thing I concluded from this one, all the microbes, all the gut microbes I found from the honeybees in Kenya, they are similar with the microbes which were reported from other latitude or Europe or US. So and what the difference we found here. Now, as you can see from this figure, the first three dominant, the abundant microbes are the Glamella, Snogelsella, and the Freshella. This Glamella and Snogelsella, they are common, even they are abundant in other latitude. However, when we look at in Freshella, the Freshella is highly abundant in our samples in Africa, in Kenya. However, it's not abundant in other latitude. And And also the lactobacillus species, Lactobacillus firmi-5 and firmi-4, we expected it is a dominant microbes in our samples. I mean, based on our information from the other continent. However, in our sample, it's not like expected. It's less abundant when we compare with others. In addition, the Commensali is also most prevalent abundant in our samples, but it is not abundant or the most prevalent in other latitude. So this may be connected with the different factors, which I'm going to highlight in the discussion part. And then based on this one, I asked myself, okay, since the general composition associated, we find this general in the composition, the different one from other, what about the diversity? What about the diversity? So I use alpha diversity and beta diversity to see if there is any variation of the gut microbe from these from the honeybees. And the my alpha diversity analysis showed there is no any bacterial community variation among those locations. That means most of the microbes from the honeybees are conserved, as I mentioned in my introduction. They are conserved in when we look at the evenness diversity, there is no any significant difference, and even the richness through the Shannon diversity and the phylogenetic diversity and the richness through using the Chow diversity, there is no any significant difference. In addition to this one, the beta diversity also showed there is no significant difference of the bacterial community variation among the location. So that means show that these microbes are conserved across the samples, even so you collect from the different regions. Um, However, we look at the one of the species, the most dominant one, the Glamella genera. It is varied across the region when we looked at it. We did the absolute quantification from our the same samples, and we found that the Glamella abundance, the highly abundant in the coastal region when we compare with the central location and the western. And based on this one, we raise some question what maybe the environmental factor is there, the environmental issue is there. So the first thing our analysis is that we correlated with the altitude. And when we correlate with the altitude, we found that Glamella is negatively correlated with the altitude. Again, we also correlated to the humidity in the coastal region. Again, the Glamella is positively correlated with the humidity in the coastal region. So the reason why this factor is happening, I may take this give you a brief in the discussion part for this one. So that maybe there are different reports on the glamella and also on other microbes. The abundance of the glamella, as previous report mentioned, uh, it can be influenced by the seasonal variation. And in addition to that, it can be also this glamella is increased. It can be increased in honeybee that are chronically exposed to the different insecticide and herbicide glyphosate, according to the few report we have seen. And the reason why glamella increases in the host when they are exposed to the pesticide is this glamella plays role to support the host for detoxification. It's break down the toxic compound. We when the bees go to the flower in the foraging, they can get the forage, the pollen or whatever with the toxic compound, like the compounds from the herbicide or insecticide. The glamella helps them to detoxify them and to break down. So that's why when there is such factor in their body, it increases in abundance in their system. So this is the important of this species. And as we find out, we do a survey information, literature, and other information. Most of these herbicide and insecticide are registered in Kenya and they are used in Kenya, especially even in the coastal region. That's what we found. The other thing, I'm not going through all the discussion, but I wanted to also highlight about the Freshella, why the Freshella is abundant and how is showing the abundance in this region. This Freshella is very important. It's important bacteria in the host, and it helps to stimulate the host immune system. And in addition, it helps also to protect the bees from the Nosema sarana infection. And that means maybe this can raise another question, another hypothesis. Okay, if the Freshella is supporting the immune system, and also maybe if the Freshella is supporting the protecting the bees from the pests or pathogens, there's maybe raise a question. So it can be correlated maybe. Okay, our bees, the African continent bees, are more resistant to these diseases and parasites. Maybe further study needed to understand this thing is how the Freshella is correlating or interacting with other pathogens in that region. To conclude this study, the study has revealed the presence of the core honeybee gut bacteria in Kenya, the same with what we found in worldwide. Oh, sorry. I don't know why I went there. Okay, good. Thank you. Um, Again, the most core bacteria in this study from the honeybees are the Glamelas, Nogricella, and the Freshella species. And then the highly conserved bacteria from these studies are, as I mentioned, in the result part, the Freshella and the Commensalibacter. And Lactobacillus is less abundant than we expected. And the study showed the major bacterial community variation among the didn't show the no major bacterial community for others. We have seen only one bacteria is varied across the region, which is the Glamella and most abundant the coastal region. In addition, we found one bacteria which is Apibacter, which is prevalent in our samples, but rarely reported from the Apis mellifera. This genus is mostly found in the Apis cerana and Apis dorsata. So in these samples, and it was prevalent in our samples, and maybe that's something which need to be studied to understand why we found this Apibacter in the Apis mellifera. So my next presentation is on the stingless bee species. What I've done so far on the stingless bee species. Um, for the stingless bees, the same question I raised for the honeybees also raised the same question. Since we don't have information about the microbes associated with the species in Africa, and I raised the same question: what kind of bacteria is associated with the stingless bee species, especially I forgot in Kenya. So for the sampling, we we sampled the stingless bee species, uh, which was uh, which was sampled by our institution from different parts of Kenya, and then we kept in our apiary in our milipulmonary station in Isipe. So we have different species. Like for my experiments, I targeted 8 stingless bee species. These 8 stingless bee species were kept here in this apiary, and then they were there at a minimum about 6 months together there, but they were collected from different origin, different sides of Kenya, but they stayed here for more than 6 months. So I collected different species. Stingless bee species are it's not like honeybees, their size, they are very tiny and small, and even it is very hard to dissect them to get the guts. I tried my best and I dissected them to get the guts for my analysis. So for this one, I did the same thing, DNA extraction and sequenced the 16S before in region, and I analyzed the diversity and abundance. In addition, since I found the kind of the variation, I also moved to do kind of phylogenetic for these microbes from different species of the stingless bees. So this is the genera or the species, the bacterial species I found from different bee species. On the left side, you can see the Afrotropical stingless bee species, their phylogenetic tree. So I connect their abundance to their phylogenetic tree to see how they are varied, to see how they are distributed and differently across the different species. So all of them, 8 species from this result, they have different composition, different abundance of the microbes. The microbes are not conserved across the species. They have a different abundance, different diversity, and different composition. Even each of them, they have their own dominant bacteria. each of them they have dominant bacteria. For example, if you look at the first species and the second Hypterogona species too, the acetolactobacillus is a dominant one. And if you can look at each of them they have a different genera and they have different composition. So from this one, since they are varied across the species in composition in abundance, I was asking myself, what about if we check each amplicon sequences or each strains in different species? And this number you can see the code as given for, like, from bifidobacteria species, we have different strains. From that one, like if you see number 4 is one of the strain, which is highly abundant in Hypterogona species 1. But when you look at the Hypterogona species 2, they are most closest species, as you can't find it there. Even if they are very close and similar species, they have quite different relative abundance. The same thing for Acetobacteria family. If you look at the different strain, like for example, strain number 2 from this Acetobacteria family, it is mostly dominant in in the Bocande, but when you look at the other related species, you can't find that abundance. Not only that one, from the other lactobacillus species, try to look at it if the strain level, species level, how they are varied across the different species. And if you look at, for example, the number one, and it is highly abundant in only one species, in Melipona toganses, but when you look at the other frigeni, you can't find that, or less abundant there. So that is interesting and showing the variation, different variation. You can see also in Bombilactobacillus species, these are the most dominant families in this species. And I was even went down to the species, the strain level, how they are varied, and they have different abundance. So based on this one, I again question maybe what about the diversity of these bacteria across the species? So I found the alpha diversity varied across the single species. That's what I was expected since the composition is varied across the species. What about the alpha diversity? So this alpha diversity, if you look at it, they have a different diversity. This one is a richness diversity on the Shannon matrix. Across the species, 8 species, they are varied, they have different diversities. While these species are from the same place and they keep the same place, they are flowering from the same place. And again, if you look at the evenness diversity, they are varied across the different species. Again, if you look at the diversity, phylogenetic diversity, phase diversity, they are varied across the different species. Again, I also looked at the beta diversity, what the beta diversity looks like. From the beta diversity, the PCO precursor distance shows the sample segregation or clustering. If you look at this one based on the species, they are clustered separately and independently here. So, and but I didn't find the kind of clustering for the unifrax distance analysis for the weighted and unweighted one. Now, uh, since these microbes, the composition, the abundance, and the diversity is varied, and also the abundance of the different strain is varied across the species, I was interested to look at it, what the phylogenetic tree looks like from the most dominant family. And then the first family, I did the phylogenetic tree for the Lactobacilli family. I took the first different strains from this one and looked how these strains are clustering with different microbes from different stingless bee species. Even here, I included the microbes from the honeybees and from the stingless bees to see how our strains are clustering with others. Again, I got the references species or strains from different continents, not only from Africa, from different continents. If you look at this one, the most amplicon sequence variant were closely related with a strain from Lactobacillus firmicus 5 and also Bombilactobacillus. However, most of the multiple amplicon sequence variants are related with Acetylactobacillus and Apilactobacillus. If you can see, and one of the ACV is related to the legal lactobacillus with a very low abundance. However, if you look at the most of our the strains, they are separately clustered. That means when I compare with other continent, our strain from the single species are uh, se- clustered separately, even when we compare with honeybee species and also from the with the stingless bee species. In addition, I did the second dominant one, the Bifidobacteriaceae family, and I checked them and looked at them how they are clustering with others. And so from this one, we found the cluster into subgroups, and the most numerous abundant were related to this Bifidobacterium commune and also Bifidobacterium vermicum and Bifidobacterium bombi. Then only the two amplicon sequence variants cluster with the Bifidobacterium corniforme. So these three, it has a short branch when we compare with the Lactobacillus, and also some of our species, our strains are clustering separately. The third one, dominant Acetobacteriaceae family, is here. I also showed this one how they are clustering with others, and the most abundant is umbilical sequence variant is related to the Acetobacter species, and only one group is related with the Bombella apis and the Bombella intestine, and the other groups are related with Nicomotomata thailandica. So from this one, what we can see from three of them, most of the strain which we characterize from the African stingless bee that clustering separately, that means there's a kind of a very novel species or strains which are associated with our different stingless bee species. And for this one, we found, I wanted to highlight a little bit, we found the phylogenetic diversity revealed the novel diversity of the strain from this group that have not been reported in other studies on stingless bee species. Despite they are sharing the same habitats for at least 6 months in the area in our apiary, each stingless species are dominated by a certain number of the specific bacterial strains. Uh, Here now, the bacterial diversity varied across the stingless species may be because of different factors. Like, for example, a study reported that the bee size and the bee colony size are positively correlated with the bacterial diversity. In our study, we tried to see how the bee size correlated with the gut microbiome, but we didn't find the correlation. But to analyze for the colony size, since our stingless bee species in Africa is at the infant stage of the study, we couldn't manage to get the colony size to correlate with the microbes. The most interesting thing here in this study that we found the endosim bound, the Wolbachia, in two species, which is 100% prevalence in Litrogona species and the Lindeliana. Um, we maybe there is something why this Wolbachia is associated with this species. We know that the Wolbachia is most important in protecting the host against the viruses. We know that there are a lot of viruses, different types of viruses, which are characterized from the African bees, which may be Wolbachia is helping, which we don't know. It needs further studies also. And also, Wolbachia is very important to manipulate the host reproduction by inducing the parthenogenesis. uh, as we know, most of those English species workers contribute to this production of the haploid eggs, which is generate the male offspring or trophic eggs that can be eaten by the queen as a food resource. But we don't have any information on this one, also need to be studied. To conclude these parts, from this study for stingless bees, we found that the Firmicutes, actinobacteria, and the Protobacteria phylum were the dominant and conserved phyla across the stingless bee species at the phylum level, while they have a varied across the species based on the genus level. And however, there was a significant variation among the genera that dominated the gut microbiome of the stingless bee species. Again, the phylogenetic analysis showed the important bacterial strain diversity, which is shared with other plate bees, but also numerous in African uh, specific bacterial clusters. So these are my two finding or research conclusion, and I want to summarize these studies as two honeybees as stingless bees. Summarize, in all the honeybees, we found the microbes which are associated with the honeybees, which are reported elsewhere in the world, and the most dominant genera in the honeybees are the Glamella, Snogracella, and Franchella, while the Glamella is really varied across the location and also correlated with the humidity and altitude. In addition, in honeybees, we found the lactobacillus was less common compared to the temperate region of the world. And in stingless bees, we found that the Firmicutes, actinobacteria, and the Protobacteria were the dominant and conserved phyla. And we also find the significant continent-specific and host intraspecies diversity in each stingless bee species. So based on these studies, I have a few recommendations. These studies establishing the base for the future studies to understand the diversity and the fluctuation of the abundance, the core gut microbiome, which are in different locations, and also it provides the foundation for the possible expansion of these gut microbiome studies in other subspecies of the Africa or in the tropical region. Again, stingless bees, as you have seen, the gut microbiome varies in composition and diversity among the host species, and therefore this study needs to evaluate the co-evolution of the gut microbiome. How's the microbe? Since the different species of the stingless bees, they have a different microbes. It needs to be studied how they are evolving with it differently, how they are interacting with it, what the role, what the benefit they are playing in these stingless bees. And this study based on the 16S analysis, which the resolution remains limited. You can't find the high resolution. So the metagenomic analysis is very needed to understand, to get a more understanding of these bacterial communities, their diversity, and also their metabolism benefit for the hosts. Finally, I would like to take you through my current project, which is only the two slides for my two projects, the current project. And my first current project is still on the honeybees, but in different ways. On honeybees Apis mellifera, but I focus on the evolution side. Now, and we know now the microbes which are associated with the bees, either it is in the northern hemisphere and tropical region, we know. But now the question is that if they are benefiting bees, if they are contributing in the metabolisms, if they are contributing in the in immune system and protecting the bees from other pathogens. So now, if you want to use other kind of other probiotics, what's the evolution? How they are evolving with this their hosts? Like for example, are they contributing only on first generation or second generation, third generation? So far, I've done sixth generation evolution, which takes about one evolution takes more than 30 days or maybe 20 to 30 days. And so I did so far the six evolution for that one to understand for this from this evolution, each evolution, I analyze and understand how they are influencing the survival across the evolution in evolution one, in the generation one, generation two, generation three, how they are influencing the host survival. Not only that one, how the bacterial, I also analyze how the bacterial abundance and diversity is uh, reflecting in different generation. Again, also understand how the diversity using the alpha and the beta, how the diversity is showing across the generation. This is my current first project, which is ongoing. And my second project is, as I mentioned in the introduction, bees are being affected with different factors, and the pesticides is one of the factors that are contributing in the in colony collapse or in population decline. So here I'm interested to see how the herbicides can affect the gut microbiome. Not only the effect, I'm also interested to see how the compromised microbes by the herbicide recover. How do they recover in the newly emerged one, in the germ-free bees? So for this one, as you know, bees are exposed to different herbicide, pesticide in different ways. For example, the glyphosate, the herbicide we use uh, in our field, in our farm, is The plan is to target the herpes, but here now this can contaminate even the water. The bees, they just fly and get the water from the environment. It can contaminate the water, and then when they get the water from the field, from the area, they come back to the hive, they contaminate the hive. They can even transfer to the other bee communities. In addition, this herbicide can contaminate also the flower. Whenever they go for foraging, they can pick up the contaminated pollen and bring to the hive. And it can even pollute the environment, the air. When it pollutes the air, it can also affect the bees recognition, or maybe if they are affected, they can't recognize, they may remain outside without coming back to the hive. That can affect the bees directly or indirectly. Directly, it can affect the host. Indirectly, it can affect the biological component like microbes, which are playing a role in bees. So I'm interested to see to rear, do the cage rearing. And the cage rearing, I expose the bees to the glyphosate. Once I expose, I see, I do the recovering of the microbes in the older bees. Once I recover the microbes, I transfer the compromised or the unhealthy microbes to the new generation, to the newly emerged bees, see how the microbes recover. First, I see the effect, then after the effect, I see how they are recovering the newly emerged bees. From this one, I'm going to get also how, while we are exposing to this herbicide, how this herbicide is affecting the bee survival, and also how the herbicide is affecting their body weight. If they are not producing a lot of metabolites, if they are not kind of healthy, it may reduce their body weight. And in addition, I also look at the microbe, how the microbial composition and diversity and abundance. Not only this, and finally, I will also look at the metabolites. I measure the metabolites and from individual bees, see and how this herbicide is affecting. That's all my project, and I would like to acknowledge the sponsor, the fund, and for my PhD fund, the DID from German government, and also Swiss National Science Foundation, Newton University Fund, and my host institution is CIPE. I did hold my PhD work, as Abdullah mentioned in the introduction, and also my host university, my University of Pretoria, where I got my PhD. And then there are different donors which are directly providing the financial support to ICIPE. That means indirectly I was supported by the different organizations. I would like to acknowledge them. And the current institution, my funding for the postdoctoral fellow, and my postdoctoral fellow is are sponsored by a National Research Council, and also my host university is the North Carolina State University in apiculture program. All these works, it was not done only by me. There are many people behind this work, this research. And first, at the Africa Reference Laboratory for Bee Health in ICIPE, there are different people here supporting in different ways. Some of them are playing in lab, some of them are playing in the field, and some of them are playing a role in the bound formats. And also my current institution, the Tarbi Lab, different people are contributing in different ways in lab and also in the field or in the bound formats side. Finally, this me. This is my address. If you are anyone is interested in collaboration and sharing information, asking question, you can reach out to me with this address, or my email is here. So that's all my presentation. Thank you so much for listening. I would like to hear from your feedback, question, comment, suggestion, because your feedback is very important for my future work. Thank you so much.
Once again, I'd like to express my gratitude to you, Dr. Yusuf, for being here today. And thanks for your informative lecture.
You're
You can hear
welcome.
me?
Yes, I can hear you.
Anyone
Thank
have
you.
a question? You can write in chat room or raise your hand, and I will allow your mic. Actually, I have a question, Dr. Yusuf.
Okay.
I can start. Um, can gut microbiota also help honeybees and the stingles bees adapt to different environments?
Abdullah, can you repeat your question, please? Thanks.
Uh, can gut microbiota also
Mm.
help honeybees and stingles bees adapt to different environments or different locations, regions like Egypt and Kenya
Uh,
can
it,
adapt?
that's that's a nice question. That's a great question. Thank you, Abdullah. Uh, for both honeybees and stingless bees, their abundance, maybe the composition can be affected by location, by different geographical location. Mostly for the honeybees, they are conserved, but due to the different factors I've mentioned, like kind of pesticide in whatever we are putting in agricultural systems, is affecting their abundance, even the microbes which are associated. And so that can make a issue. Like I mentioned, in the coastal region, we find one is most abundant, the others are less, like lactobacillus is less. It's because maybe for different factors, as I kind of mentioned in my current project, looking for the herbicide effect. Herbicide really affects, like for example, glyphosate affect the gut microbiomes directly. It really targets a specific enzyme which it manipulates and kills the bacteria. That means indirectly affects the bees. So different factors can affect their abundance from different region. Yes, even as I mentioned for the microbes correlated with altitude, even the altitude can in different location, different humid can affect the microbial composition and even season can affect.
Okay.
Let me know if I answered your
Uh,
question.
Yes, yes, Dr. Yusuf. We have a question on in chat room. Uh,
Mm.
Dr. Islam Muhammad asked, is there a correlation between food consumption and gut microbiota?
Yes, the answer is yes. It can really affect. And the
Okay.
reason why, to explain a little bit here, uh, as I mentioned, in honeybees, we have about 9 phylotypes. 5 of them are very specific to honeybees, and 4 of them you can find anywhere. Look, the one which are kind of related with the environment, when they go to the flowering, they can rest on flower and get all the pollen, come back to there. They can bring even the microbes from the flowers to the hive. Again, not only that one, that's a direct. Indirectly, when they have sufficient food, it supports the microbes abundance and composition and in the survival in their body. But when there is a kind of shortage of food, that can directly also affect the microbes. Once it affects the microbes, indirectly it affects the host. To summarize this point, the food consumption can affect the food, even the food, different types of the food, the amount of the food can affect the gut microbiome in both.
Okay. Someone asked Dr. Muhammad Slim asked, if the correlation is negative or positive between them.
That means with the food, right?
Yes, I think that.
Let me add on Dr. Muhammad before I answer this question about the age. That's also a very interesting question. Yes, the age, there's age factor. Like for example, whenever we characterize the microbes, we need to focus on the specific age. If you are, for example, if you are like, for example, when bees you see the newly emerged one, it takes them 5 days to completely colonize their gut with microbes. Bees, the way they transfer microbes is very a different way. They interact together, they work together in the hive. So when they interact to each other, they transfer bacteria to each other, or they can be inoculated from the hive. That means it takes 5 days to get the 100% colonization. So if you analyze the microbes from 2 days old or 3 days old, it's going to be the same with the 10 days old bees. That means age directly affects. With the correlation, I don't know if is it correlation with the food or with the age. The second, the last, this
Mm.
question.
Dr. Muhammad Islim, can you explain that? He writing now.
Dr. Islam is raising his hand.
Yes, I will allow. Yes, Dr. Muhammad Islim.
Yeah. Muhammad said the microbial gut and the consumed. Okay. Yes. The correlation, like if they have sufficient food, nice food, and good food, the correlation is positive. If the food is bad, it can deteriorate their gut microbes. That means if it is good,
Okay.
it's positive correlation. If bad, negative correlation. I hope
Okay,
you got my answer.
I will allow your mic, Dr. Islam. Dr. Islam, please, you can unmute your mic.
Hello, hi.
Hi, hello.
Hi, Dr. Islam.
Hi, Assalamu alaikum.
Alaikum
Walaikum
assalam.
salam.
Okay, Islam Omar, Asit University. I just wanted to ask about a little thing about the gut microbiome. First, you said in your new research that there is like a correlation between the humidity of the area and the gut microbiome, or the abundance of the gut microbiome in this area, right?
Yes.
Okay, is it the bees you examined? Is it the same age and the same colonies and the same everything, or is there a difference between them?
That's very nice question. Thank you. The age, yes, I target the adult age, the forages. We
Okay,
have different types of stay age, like kind of some of them are nuts. They don't come out, like they come out after 20 days to go for the foragers. So
okay,
I focus those people are working outside because they are the one who is investing a lot. To tell you
foragers.
that, yes, the foragers, and I close the
Okay,
hive. I close the
yes.
hive and I collect the one with the pollen. That means the one foragers the same age.
Okay, nice.
Yeah.
Okay, you didn't examine the food or the bee bread inside the hive, or maybe
Uh,
correlate the bee bread and the microbiome in the midgut of each area.
for my study, I didn't, but before my
Yeah.
study, a lot of things done in the other continent, like in Europe, in the United States. People have done that one
Okay.
correlated with the food. They have seen the correlation. A lot of information
Okay, but in this study, no.
in this study. No.
Okay, there was like a difference in humidity in the three areas studied, and there is
Mhm.
a correlation between the humidity and the gut microbiome. Is it right?
Yes, there is a positive correlation in the coastal region with the humidity.
Okay, another and the last question: is it the same plant coverage in the three area is the same, or it's like a difference between the area plants in these three areas?
In in Western Kenya, there's agricultural farm, and there's a kind of Western Kenya, the forest is, and also in the central, there's some highland, and there's a kind of we have a small agriculture. Some of areas are urban areas, and in the coastal region, we also have a small farm areas. A little bit, yep. The coastal region may have a different vegetation, but there's still a farm.
Okay.
And that as yeah, I understand your concern. The vegetation may affect. Yeah, it needs
Yes.
also further study. Like for
Yes.
example,
Yes.
I mentioned different factors like maybe this variation comes from the pesticides or which we are using in agricultural areas, or maybe the vegetation. That means it need a kind of deep studies, like kind of finding out, looking at the vegetation plants and looking at the herbicide, all the
Yes.
agricultural input we are using. So I understand that concern.
Okay. Okay.
Yeah,
Thank you.
you're welcome.
Thank you, Dr. Islam. Any question? Anyone have question? Uh, I think no. Okay, that's in, Dr. Yusuf.
Thank you so much for hosting me. Thank you for so much for giving me the opportunity to present my work. And we shall keep in touch. And anyone who is interested in this area, please reach out to me. Here's my email. In case you need any
Okay,
question or you need help,
okay.
you're most welcome.
Okay, thank you, Dr. Yusuf, and thank you, our audience. Please register your attendance in the form provided in the chat, and see you next week.